And we're live. Welcome to the Evidence and Reasons for the Christian Faith video channel. I'm Salvador Cordova, and I'm joined by Emory Moyna tonight. And we're just going to have a typical research session that grad students or researchers might have. I want to give the audience a feel for what it's like, and it's pretty boring and tedious and laborious. But maybe it can give some appreciation for the scientific method. And also, I think it's good to see creationists actually trying to do, not trying to do a dog and pony show, but really just kind of in their everyday life of trying to explore God's creation and to fight against evolutionary theory. And it can be it can be a very tedious and long drawn out affair. I mean, some of these creationists have been in the battle for, it seems like 50 years, half a century. And then...
I mean, some of them aren't that far away. I mean, if you think about someone like Kurt Wise, he got his PhD in the early 80s. We're talking, he's into it for 40 years. I mean, some of these guys have been around for a long time and been fighting this for a long... And I mean, I don't know how active Dr. John Morris still is, but... like, he's been in it for 50 or 60 years. So there are some of these guys that have been around for a really long time.
And the importance of that is that, you know, sometimes on the internet there's a desire to have quick victories, and that can be affected with kind of the personal charisma of the people on YouTube, and then maybe being, unfortunately, pretty loose with the facts and not being thorough and only giving one side. For those of us who do this professionally and also care about students going to school, we feel obligated to tell it as good as, you know, we could say we don't believe this, but, you know, this isn't a done deal yet. We don't have quite as many facts as we'd like in hand. Yeah. And so I get...
to be perfectly honest, what scientist has ever said, yes, we have all the facts we want?
I can't remember any.
Exactly.
But you know, this is very different than what you hear on Sundays and from the preacher. And I mean, because the people that know me, the Christians especially, have always thought I was an atheist because I'm just as skeptical as they are. You know, I ask hard probing questions, and I'm really not impressed by too much fluff and argument by assertion. I'm just not.
Yeah.
And it's caused me a lot of difficulty in the churches, just trusting what I hear from the pastors being true. That's just my nature. You can't be a good engineer if you believe everything you hear. I mean, certainly if you're in the procurement process, you better not believe everything the salesman tells you. I mean, come on. So, I mean, it's just been very hard. Now, ironically, it has served me well because of some of the churches I've been a part of that I've not really bought off on the pastor's character or what he's saying. I mean, I don't like to treat people that way. I would love to meet people I totally trust and believe in. But then I just look at myself. It's like, Sal, do you really trust yourself to do everything you think you know you should do? And just like, no. You know, I had my plans for what I was going to do this, you know, in the morning. It's like, yeah, I don't know that I even got half of it done. So, you know, I'm not trying to be openly hostile and distrustful, but we just kind of know what human nature is. So, I guess it's okay to say, you know, I might be on an upcoming show. The time hasn't been solidified, and I'll be in an environment, I might be talking about yet curriculum. And I never even thought about that because I don't have any kids of my own. And I don't know what to tell parents what to teach kids, because at the professional level in creation science, it's like, yeah, we believe in Noah's flood. Many of us believe the cosmos is young. But then what do we tell their kids? That is this all solved? Because the reason we have professionals, researchers at this, is there's still a lot of outstanding problems. And so this has given me some pause for thinking. It's like, how do we present a balanced view and yet also convey what we believe? And that's just a question I never thought about. Although, as John Maddox said, every now and then when I have doubts about, like, say, the basics of the existence of God, I just look at the cell. And like every day, like when we have shows like this, and we actually just go through secular peer-reviewed papers or textbook articles, I'm like, yeah, maybe the chronology of events is still contentious, because I say ID is easy, YEC is hard. Being able to do clocking, and we're going to talk about clocking today. And I was just reviewing an interview I had with Stephen Meyer, and he said, ID is not really concerned about the when and how. It just says it's designed. Whereas, especially the young Earth creationists, we're concerned with the when and how. How is in like, what were the mechanical, naturalistic mechanisms involved in the flood and formation of the geological record. And whereas we believe the creation of life was miraculous events, like, no, we're not even going to try to frame this in terms of repeatable laws of physics. But Noah's flood, radiometric dating, molecular clocks for like mitochondrial Eve, Y chromosomal Adam, distant starlight, we're actually looking for naturalistic mechanisms, and that makes young Earth creationism really hard as a research enterprise. Do you have any thoughts, Emory? And I'm going to acknowledge.
I think that's mostly spot on. My some of you may be aware that I have a teaching background. I actually taught in a Christian school for a couple of years, and so like I kind of got a taste of dealing with the problem of what do you teach someone who is up and coming as a creationist. What do you teach them? And so it's interesting. And there are questions where, yeah, we probably have this pretty well resolved, but there are other questions where you kind of go, man, I don't know. Now, most high school kids really aren't interested in diving in that deep, just being real. But the few that are, you sit down with them and you go, okay, I got answers for this. I got answers for this. I don't know what to tell you about this one. Like, we just don't have a complete answer for this yet. And I mean, and that's why Sal and I exist, to try and come up with some of the answers for some of the questions. I know those of you who may have watched the shows from David McQueen on a different channel, he likes to say that when Henry Morris founded the Institute for Creation Research, he didn't found the Institute for Creation Answers. He founded the Institute for Creation Research. And yes, we're going to get some answers as we do some research, but there's always going to be questions that are going to be open and need to be asked. And that's something that I think is valuable, being able to dig into the meat of things and try to answer some questions like we're going to do tonight.
I got to tell you, I mean, I'm rarely on the same page as Kurt Wise on a lot of issues. But his persistence is inspiring. Yep. And I think what I would tell the younger generation, and the question is, at what age do you start to tell them the other side? And this is a discussion I'd love to have with Nathaniel Jeanson, because I asked him, how were you raised up? And how is it that you were able to remain a young Earth creationist? And
hey, Sandy, how you doing, man?
Hey, Sandy. And he said he was taught the other side when he was homeschooled. I was very impressed by that. Because some parents think, oh, we can't expose them because they'll lose their faith. I'm just like, okay, once they grow up and they leave the nest, they're going to be exposed. Would you rather have them hear the other side while you're there to talk to them about it, or you're just going to let them go free and they're going to be just shocked? They'll hear stuff that they've never heard.
Yeah.
And go on.
For me, I think that and this kind of gets into the education stuff that we may talk about tomorrow night, because spoiler, Sal and I are both going in and sticking our heads in the lion's den. But And by the way, if anybody who watches that who's going to be related, that's just a metaphor. I don't actually know that much about where we're going. So, but what I was getting at was, I think there's a time and there's an age where you start introducing them to arguments that are opposed to the faith. I don't think you give that to them in kindergarten, but maybe as they get a little bit older and they have a little bit, and each child probably is going to have a different jump off point for this, exactly. Right? Because some kids, you can probably start them in second grade, right? It's because they're real smart, they're ahead of their game and all that. Other kids, maybe particularly kids maybe with a learning disability, you may have to wait a few extra years because they need a foundational grasp of what they're doing before they have to be introduced to a concept that is a challenge to what they already understand. And so it's going to differ, but I don't think it should be a side-by-side thing. I think it should be, okay, let's take some time at this point here, and whatever point that is, and particularly if you're homeschooling your children, you can make that decision. Which, by the way, homeschool your children. Sorry. I'll be controversial. Let Sal be the nice guy.
No, teach them that racist mathematics,
2 + 2 is 4.
Yeah, that's just so racist,
Anyway, but particularly if you are homeschooling, you're in a small Christian school, you can kind of point to, okay, here's where I want them to start learning about this because I think they're ready for it. And if your school doesn't offer some kind of apologetics training at some point, if you're in a school setting, you may want to look for another school that does. Because, and I know that because I taught in a Christian school, and I offered before my second year teaching, hey, let me design an apologetics class. Let me for the seniors and juniors. Let me design an apologetics class. I'll write the curriculum. I'll do all the work. And I was offering to do a ton of work, which, and they didn't want it. They didn't want it. So it's kind of like, which was
no surprise. I'm sorry. I mean, no surprise, and that's been my experience too.
Yeah. Yeah.
And,
but I mean, that was
go off to college and they'll be slaughtered.
Yeah, I know. And I think most of them were. But that was kind of one of the straws that broke the camel's back when it came to teaching. It was kind of like, okay, yeah, that's the end of that. I'm offering to do an awful lot of work that will be very beneficial to these kids. And I wasn't just going to cover creation, although that would have been a significant part of it. I would have addressed some other issues that are prevalent in the culture. This was 10 years ago, so culture wasn't quite as disastrous as it is now.
Well, yeah, go on if I interrupted you. Sorry.
No, I was just going to say, but they wanted no part of it. So anyway, go on.
I was going to say, prior to the fast information age, the World Wide Web, where people had such easy access to opposing viewpoints, and then also when the universities were at least a friendlier environment than they are now toward the Christian faith. The business model is, let's just do indoctrination.
Mm-hmm.
You just protect them, quote unquote, brainwash them. You don't really teach them how to think. And that might be good enough to get them by. And as long as they don't have a deep crisis, they might stay in it. I mean, that may be harsh, but the reason I say that is, why wouldn't they want apologetics? It's like, oh no, just repeat. Don't expose them to, why teach them how to defend their faith? Just kind of, program them.
To be fair, that is most curriculum in general, public and private. They're not, the questions, particularly the test questions, are multiple choice, fill in the blank, short answer. They don't have to actually think. They regurgitate things from the textbook. And by the way, this is not just a Christian problem. Like this is a education-wide problem. And I got frustrated with it about halfway through my first year, and I said, look, can I write my own tests? Because this is ridiculous. They're like, all they have to do is memorize the textbook and they'll get an A. Like that's not, they don't have to think. They don't have to understand what's going on in the classes I was teaching. And I was told, no, we have to keep the tests the same to keep everything standardized. Which, okay, I understand why you want standard, every teacher to be, because we were all using the same curricula for different classes. So I understand why you want that standardization. Okay, I understand that. But to me, I don't want you to sit there and regurgitate information. I want you to learn. I want you to understand and comprehend. Like we talk about, you know, we were talking about I taught history for 1 year. Some of you probably won't be surprised by that. You know, we talk about, you know, the fall of Constantinople. I don't want them to tell me what year it fell. I want them to tell me why. I want them to understand what was going on in the world at that time that explains why. I don't want them to tell me that, you know, chlorophyll is responsible for photosynthesis in plants. I want them to tell me how.
And you just enlightened me. I had a thought as you were saying that. There is a point when rote memorization is just really important.
Yeah.
You know, even now I have to say, okay, adenine, adenine, cytosine, thymine, guanine. All right, and then I don't even think. I mean, my biochem professor was old school. He said, "You're going to give me all the amino acids, all 20 of them. You're going to give me the single letter abbreviations, the 3-letter abbreviations, and you're going to draw the diagrams of each of the molecules." I'm like, yes, sir. And second semester, he said, you're going to draw the Krebs cycle. You're going to name every enzyme in the Krebs cycle and what it does. And sometimes that is rote indoctrination.
Right.
But the reason that is valid, this is testable.
Right.
I mean, this is almost like learning the alphabet. There's not any controversy that we call certain molecules adenine.
Right.
But
right.
The thing about the Christian faith is you will always be challenged because there'll be other viewpoints in the world. And that's where the critical thinking comes in. And so what ends up happening is they extrapolate that memorization indoctrination model into the field of where it's actually a battle in critical thinking. And it's a different mode of thought. An airplane taxiing on the runway is different than when it's flying. You need a different skill set, all right, for the phases of the mission, so to speak. And this rote memorization indoctrination has been kind of overdone. And I see it in the churches. And
right, I'm not attacking rote. That wasn't my. I'm not attacking rote memory for foundational important things like the foundational. Adenine, guanine, cytosine, thymine, you know, A bonds to T, G bonds to C. I almost said A bonds to C. But, you know, that's important, right? Being able to memorize those things. Although, to be fair, Watson and Crick didn't know the nucleotide structures when they discovered what the structure of DNA was. They had to keep looking it up, which is rather funny. But, yeah.
Briefly, Dr. Dan is having a show. We might be on there. I hope you don't mind me trying to advertise Dr. Dan, if you've not already broken the news. So greetings.
Yes. Okay. So this comment from Dr. Dan, not the one right below that, Sal. This is actually a really good kind of question to ask a student because it tests their understanding of what is going on in the process. It essentially says, okay, you've memorized this, you know, you've memorized this system or this equation. Okay, now we're going to break something. Now we're going to break something, and here you go. Right, and this is why this is the kind of question I like, and this is the kind of question I would, if I'm writing tests, I am going to put on a test because I'm going to say, okay, you've learned all this material, now apply it. Let's take what you've learned and let's apply that and say, okay, understand this and go from there. But we've rambled on for 20 minutes. We haven't gotten anything what we were planning on doing, so.
Well, I know we've rambled on. It's just because of the things that are coming up. And I really was bothered by this because the Lord commanded us to raise our children in the faith. And I was like, okay, so I really have to think about this for the next couple of days. And I need to consult people like Nathaniel Jeanson. It's like, how are you raised? I thought that was really interesting. He told me he was given both sides. because I specifically said, you went into Harvard, you went into all sorts of hostile environments. How did you keep your faith? He said it was because my parents gave me both sides. And I'd be curious to find out how his parents, every kid's unique, but I'd be curious to find out how they decided when they're going to give the kids the other side. So I think for little children, like I've heard that even two-year-old children can be taught the scriptures. They learn as you're teaching them to talk. You can teach them the scriptures. I think that's perfectly fine. They're not at the stage you're going to give them the other arguments. I'm certainly not going to read Origin of Species to them. Sorry, Dr. Dan. I know you might do that for your kids, but not for creationist kids.
I don't know. I wouldn't read Origin of Species. That's old English. You want to read them something in modern English if you're going to do that. Maybe read one of Dawkins' books or something if you're going to do that.
Yeah, Blind Watchmaker. No, Dr. Dan doesn't think highly of Richard Dawkins. So someone more palatable. So oh wow, here's a biology professor. And just for what it's worth, guys, I'm sorry I have to take another swipe at Dr. Dino here, but we did vote Dr. Dan. We had a little poll. Who would we have as our biology teacher? We as creationist students, between Dr. Dan and Kent Hovind. It was unanimously Dr. Dan. Just thought we'd let you know, Dr. So tonight, I know we rambled on, but I just had to get stuff off my chest. This is part of what we do as researchers. Sometimes we just get stuff off our chest before we do our work. Sometimes we never do our work, which is kind of pathetic. But Richard Sternberg. We could read some of his articles. I'd like to read the one he had on junk DNA. And I can drop it in the private chat since, how's your voice doing after your flu there? You're looking a little worn yesterday because I know you did have a full day of work. If you're rested.
No, I should be good to do some reading, Sal. I've got a water bottle here too. So if I may need to jump and grab another water bottle at some point, but I should be okay.
Yeah, I may need to I'm going to go get some apple cider myself. But I dropped it in the chat. Oh, wait, wrong article. Sorry.
I hadn't opened it yet, so you're okay.
Let me get... I mean, this will give you an idea. The reason I like Richard Sternberg is that he has two PhDs, one in evolutionary, molecular evolution. So he's an evolutionary biologist. And the other is in systems theoretical biology. So Richard Sternberg, PhD. And we may read a little bit of his essays of his transformation, but he was a committed atheist. Intellectually, he described himself as an intellectually fulfilled atheist. Went through the evolutionary curriculum, and then got associated with the intelligent design movement. So I think it's going to be compelling to hear also his journey. I think somewhere I read he had Catholic roots. And he said he's kind of going through postmodern anxiety. And this was like 2004. I don't know where he is now, but people have said, after he got the congressional investigation and the U.S. Office of Special Counsel, which is, I think, a presidential appointee, and after he got kind of persecuted and battered around, some people said, yeah, he's starting to probably reconsider a lot of things. And I don't know where he stands now, but the last stuff that I read seemed to be extremely theistic friendly. And he seemed to have had much more friendliness to intelligent design than when he had that article in 2004. And so anyway, I think it'd be great to read what he has to say about junk DNA and how it is used in the mouse. Now, I will say this before, if you could prep that to post on the screen, I'll have a few words, and then we can proceed with the reading. But I worked briefly I worked for about 1.5 years at the Army Fort Belvoir Night Vision Labs. So as many of you know, I was an engineer and scientist in the aerospace and defense industry. The only time that I recall that I was given the title of scientist when I was actually working at the Night Vision Labs. I was a junior scientist, and I was working in their automatic target recognition project. And that was interesting. But the whole thing is these guys were also just steeped in study of photonics and optics, geometric, just lots of physics, lots of solid state physics, photons, study of photons. It's going to be interesting because Richard Sternberg will touch on the relationship of photons and lensing and junk DNA. It's just going to be awesome. And you know, when I first read this, I did not have I was not working for Dr. Sanford at the time. But now that I have the biology background, you know, my jaw is dropping even more because I had my optics background and I was already wowed. But then now that I have my biology background, and he's describing all these things with, you know, chromosome bands and the optical properties of DNA, stuff people don't even consider. This is going to be an awesome article. So, Emory, take it away. While you're reading, since I already read the article, I'm going to grab some apple cider and I will join. I'll rejoin the stream.
All right. So here we go. Shoddy engineering or intelligent design. Case of the mouse's eye. Now, I'm just going to tell everybody, if you have comments on this, I can't see them. I'm reading it. So I'm on a different screen. So please, if you're shouting at me that I've said something wrong or whatever, or I skipped a sentence, I'm sorry, I can't see you. All right. We often hear from Darwinians that the biological world is replete with examples of shoddy engineering, or as they prefer to put it, bad design. One such a case of really poor construction is the inverted retina of the vertebrate eye. As we all know, the retina of our eyes is configured all wrong because the cells that gather photons, the rod photoreceptors, are behind 2 other tissue layers. Light first strikes the ganglion cells, then passes by or through the bipolar cells before reaching the rod photoreceptors. Surely a child could have arranged the system better. So they tell us. The problem with this story of supposed unintelligent design is that it is long on anthropomorphisms and short on evidence. Consider nocturnal mammals. Night vision for, say, a mouse is no small feat. Light intensifies during night can be a million times less, light intensities during night can be 1,000,000 times less than those of the day, so the rod cells must be optimized. Yes, optimized to capture even the few stray photons that strike them. Given the backwards organization of the mouse's retina, how is this scavenging of light accomplished? Part of the solution is that the ganglion and bipolar cell layers are thinner in mammals that are nocturnal. But other optimizations must occur. Enter the cell nucleus and, quote, junk DNA. Only around 1.5% of mammalian DNA encodes protein. Since it has become lore to equate protein coding regions of the genome with genes and information, the remaining approximately 98.5% of DNA has been dismissed as junk. Yet for what is purported to be mere genetic gibberish, it is strikingly ordered along the length of the chromosome. Like the barcodes on consumer items that we are all familiar with, each chromosome has a particular banding pattern. This pattern reflects how many different types of DNA sequences are linearly distributed. The core of a mammalian chromosome, the centromere, and the genomic segments that frame it largely consist of long tracts of species-specific repetitive elements. These areas give rise to, quote, C-bands, unquote, after a chemical stain has been applied. Then alternating along the chromosome arms are 2 other kinds of bands that appear after different staining procedures. One, called R bands, is rich in protein coding genes and a particular class of retrotransplants called signs, short for short interspersed nuclear elements. Signed sequence families are restricted to certain taxonomic groups. The other is termed G-bands, and it has a high concentration of another class of retrotransposon called lines, long interspersed nuclear elements, that can also be used to distinguish between species. Finally, the ends of the chromosomes, telomeres, are comprised of a completely different set of repetitive DNA sequences. In general, C-bands and G-bands are complexed with proteins and RNAs to give a more compact organization called heterochromatin, whereas R-bands have a more open conformation referred to as euchromatin. Why bother with such details? Well, each of these chromosome bands has a preferred location in the cell nucleus. Open any good textbook on mammalian anatomy and you will note that cell types can often be distinguished by the shape and size of the nucleus, as well as the positions of euchromatin and heterochromatin in that organelle. Nevertheless, most cell nuclei follow a general rule where euchromatin is located in the interior, in various compartments that are dense with transcription factories, RNA processing machinery, and many other components. Heterochromatin, on the other hand, is found mainly around the periphery of the nucleus. A striking exception to this principle is found in the nuclei of rod cells in nocturnal mammals. Reporting in the journal Cell, Irina Solovey. Dr. Solovey, I apologize if I've just butchered your name. And co-workers have just discovered that, in contrast to the nucleus organization seen in ganglion and bipolar cells of the retina, a remarkable inversion of chromosome band localities occurs in the rod photoreceptors of mammals with night vision. First, the C-bands of all the chromosomes, including the centromere, coalesce in the center of the nucleus to produce a dense chromocenter. Keep in mind that the DNA backbone of this chromocenter in different mammals is repetitive and highly species-specific. Second, a shell of line-rich G-band sequence surrounds the C-bands. Finally, the R-bands, including all examined protein-coding genes, are placed next to the nuclear envelope. The nucleus of this cell type is also smaller, so as to make the pattern more compact. This ordered movement of billions of base pairs according to their barcode status begins in the rod photoreceptor cells at birth, at least in the mouse, and continues for weeks and months. Why the elaborate repositioning of so much, quote, junk DNA in the rod cells of nocturnal mammals? The answer is optics. A central cluster of chromocenters surrounded by a layer of line-dense heterochromatin enables the nucleus to be a converging lens for photons, so that the latter can pass without hindrance to the rod outer segments that sense light. In other words, the genome regions with the highest refractive index, undoubtedly enhanced by the proteins bound to the repetitive DNA, are concentrated in the interior, followed by the sequences with the next highest level of refracticity to prevent against the scattering of light. The nuclear genome is thus transformed into an optical device, that is designed to assist in the capturing of photons. I'm going to pause there for a second and make sure, I'm going to reread that sentence, just make sure everybody got it, because again, I can't see the chat. The nuclear genome is thus transformed into an optical device that is designed to assist in the capturing of photons. Wow. That's a sentence we'll probably come back to. This chromatin-based convex focusing lens is so well constructed that it still works when lattices of rod cells are made to be disordered. Normal cell nuclei actually scatter light. So the next time someone tells you that it strains credulity to think that more than a few pieces of junk DNA could be functional in the cell, that the data only point to the lack of design and suboptimality, remind them of the rod cell nuclei of the humble mouse. And hopefully Sal is back. Yay. Oh my. Oh my. You're muted, Sal.
Bravo. Praise the Lord for his great works. Now, if people are wondering, they saw this briefly. that is first in honor of our resident Navy SEAL, retired Dr. Sandy Pigeon. He should know what this is. This, I believe this is a night vision setup. I mean, this is not trivial stuff to make night vision possible. And the mice have it. Just amazing. That's why I said, and it's really funny, and it was kind of mean. I was poking fun at Jerry Coyne. He was talking about how stupid the eye is designed. And some physicists, because he's talking about the backward retina and all, some physicists came out and said, this is not bad design. This is ingenious, because they are waveguides. And there was some guy in my apologetics prayer group, and he said, I design waveguides. This is mind-blowing. You could make it on that scale. I mean, you've got to engineer it perfectly. You know, I was talking about ion channels in another video. You have to have sub-angstrom precision in your engineering. There are not many machine shops that can do sub-angstrom machining. That's 10 billionths of a meter. And waveguides of optics would be in the angstrom range also. You got to have it right. And that supposed backward wired retina, the wires are tremendous waveguides. The genius to construct that. I'm just like, okay, so we hear the evolutionary biologist saying engineers don't have any business talking about evolutionary biology, and the engineers will say, hey, you don't have any business, evolutionary biologists don't have any business deciding what are good or bad designs, you know? And this whole thing about, I mean, Ken Miller, the whole gang was saying, stupid design, junk. This is not, you know, and we're getting from the biophysicists and the engineers a totally different story. It's like, this is kicking our rear ends. We can't build stuff this good. For what it does, yeah, we can send people to the moon. We can build computers that can do multiplication and addition faster than the human brain. But in the ways that we do things good, we can't even compete.
Junk DNA is optics. That is something I didn't see coming.
See, brother, you hang around me, you'll get all sorts of little treats here and there. See, now okay, this is the one, I told you I hate genetics, but this is the one time I really liked it because it was combined with biophysics.
Right, yeah.
Yeah.
Who am I?
Told you Sternberg was good. Now, I got a funny anecdote. You'll love this. Todd Wood was criticizing Richard Sternberg. And I thought that's the funniest thing because Sternberg was defending the ID position and Wood was defending the evolutionary position. So you had a creationist defending the huh?
That's standard. He defends the evolutionary position all the time. I sometimes wonder which side he's on.
But the funny thing is this time it was the evolutionary biologist defending the creationist, and then Wood, the creationist, defending the evolutionary biologist. It was the funniest thing. But Sternberg knows his stuff, man.
Oh my, wow.
He's just out of the public eye because you get the idea he's very reclusive. He is, but he's good. And
oh yeah.
I mean, how did he dig stuff up like this, honestly? And there's just other stuff he digs up. I'm just like, wow.
Yeah.
So he may not be known in the public eye, but among the quote unquote intelligentsia of the ID movement and especially the ID movement, he's like number one.
Yeah. He's even in creation circles, like the people who do the research, they know, and they know who he is, and they pay attention to when he says stuff.
Yeah, those are the old good old days of the creation, the Viraminology study group. There was Richard Sternberg, Paul Nelson, and Stephen Meyer. Those are the good old. I'm sorry to say that. I don't mean to insult the current group.
I'm well aware that it has taken a nosedive.
So Sandy did ask, can we discuss gene regulatory centers? That was one example of a very novel way of making a regulatory center. You're regulating optics. I mean, this is just, and I need to point out something. You can look up the cell type of lens, of the eye lens. There's another thing while we're talking about gene regulation. We also have organelle regulation in development. You know why the eye lens is so clear? It doesn't have hardly any organelles in it. In the development of the lens cell type, there's like a death signal that says, don't develop all these other compartments that you have in standard cells, because you need to pass light very clearly and not have any barriers. So I was just confronting some evolutionary types and saying, okay, so you have this proto-cell and it's going to start killing its own organelles. How is that ever selected for before you have a functioning eye? I mean, you know, he was expressing it in terms of like the rhodopsin and all of the stuff in the you know, signal pathway of the retina. But there's other stuff, just basic geometric optics that we just bypass. And I was telling Rebecca Davis, I said, now that we're going into the cellular realm, just stop worrying about the fossil record, Tiktaalik, the chimp human ancestor. And Sandy Pigeon kind of told me this. He said, okay, you know, you don't fight the guy in the thing that he's skilled at. You go after him in the thing you're skilled at. Mm-hmm. And I'm saying, Rebecca, just don't worry about that. Don't worry about the Lenski experiment, but we'll cover it a little bit today by an article co-authored by Lenski. Sandy said, just, you know, he was talking about like you don't fight a jiu-jitsu master with his jiu-jitsu. If that's not your skill, you have to fight him another way. The classic one was Indiana Jones, where the guy was really good with a sword. Yeah, Indiana Jones just grabs his pistol. Because Indiana Jones is about to fight him with a whip versus the sword. And he said, nah, just the pistol. So I was telling Rebecca, get into the cellular realm. You'll have all these strong arguments. And I don't know why I never, you know, when people are arguing with me about junk DNA, I keep forgetting this article. And this is the thing about Brandolini's law. How long did it take us to read through that? About 10, 15 minutes carefully?
Well, it wasn't even that long.
Okay, maybe 10. In a debate, they're just not going to bother. Mm-hmm. You know, the other side can, you know, if it were Matt Dillahunty, he'd interrupt me in the middle of it. I don't have time for you to lecture me. I wanted, you know, I wasn't quick enough to recognize that was a pretty slimy move on his part, because the debate, as far as I was concerned, was not to elevate either of us, especially him, but to serve the audience with information they may never have heard yet.
Right.
So that's why sometimes in these debates, you would not be able to get to this level of depth to talk about junk DNA. So what are people resorting to? They'll just quote one little sentence quotes from peer-reviewed paper, and the other guy will just find other sets of quotes, and nothing's ever resolved. That's why a show like this is just a lot of fun. Right. We can get to the real meat of things.
Right. And this is part of the reason why I haven't really done any debates, because I just like, unless you can give me an opponent who is like not just there to like play showboat, it's not worth my time. It's not worth anybody's time. We want to actually dig down into the issues, not just trade points at each other's expense. I'm just not looking at, anyway, go ahead,
Sal. Sandy was asking if this is the perfect refractory element. I don't think so, but it's pretty good. It's pretty good. I think no one has actually studied, except maybe the only people that have studied the optical properties of DNA would be the Illumina and PacBio, who build the sequencers, because they use lasers, which is interesting. They have to know, like they can tell what nucleotide it is, I think, by the laser beam. So it does have optical properties that are important that we do leverage. But as far as being the perfect, that's a specialty of biophysics that it's not my specialty at all. I actually have no specialty. I'm just so new to the field. But this was definitely an example of biophysics. It's just totally cool.
Yeah, that's wow. You don't hear you'll never hear that, folks. Those of you who are thinking about going into graduate, you will never hear that from your graduate school professor, unless you were actually taking biophysics classes. That's the only way you'll ever hear that.
Yeah. So I hope this kind of made up for last night. It was, last night kind of dragged. So at least I gave you a gem right here. So I started off with something good. I can give you another Sternberg. See if I could find another Sternberg. Well, we actually covered it yesterday with the mouse and the extrusion loops.
Yes, we talked about that yesterday.
So for those who saw yesterday, I gave a video of the chromatin extrusion loops and the signs in the CTCF binding site. Okay, so I'm sorry, this is just so nerdy, but so you have the junk DNA in one case being an optical lens in the eye. But then in other cases, the other forms of junk DNA, quote unquote, like the signs, were used to make these form these three-dimensional loops that are important for the regulatory centers. That's what really sets things apart. And there's pretty good evidence that these cannot be randomly just strewn over there like Swamidas was saying. Sorry, can't help it. Take every opportunity to take a swipe at his advocacy. Oh, evolution's a great scientific theory. So, in one context, it's a great lens. In another context, it's regulating these loops. But the loops are regulated differently on cell types. And as you saw it here, the euchromatin and the heterochromatin are, in one cell type, it's one way, in the other, especially in the lens, but in the other cell types, they're the other way. So you have these higher order chromatin structures dependent on the cell type. And there are thousands of cell types. In fact, one could reasonably argue every cell is kind of its own type. I think Francis Collins said every neuron is different from every other one. We're unique. How can you do all the accounting for that to build something as complex as the brain?
I'd like to see the pop gen that allows that to happen, Sal.
Okay, so we started off with the wow. And let me see if I can grab richardsternberg.com. And I would just like, see, now that we've had the privilege of hearing some, that was one of his popular articles. Now, I've read some of his peer-reviewed articles, and they're almost impenetrably difficult. So he knows how to switch gears for the popular press versus the academic press. But you could tell there's just a great mind, and just a great mind of a researcher, that he knows how to look for this. So, but I would like, we could perhaps do a reading of his, see if I have it here. I found it. Okay, here, I think. Okay, I'm going to try to dump this in the private chat, and it should be a document that's a PDF.
Okay.
And if it doesn't, I'll give you a link, and I'll tell you where to look. Thanks for that reading, by the way.
Sure, no
You're
problem.
saving my voice.
No problem. No problem. Let me see if that's popping up here. Ah, yes. Yes, okay. So, oh my goodness, this is 12 pages. All right, yeah, I've got it, so I got it. You want me to read this?
It should be his transformation of views or something.
Yes, that's what I've got.
Yeah, let's do it. I think this would be just great to hear.
All right, this is 12 pages, so this is going to be fun.
How about your discretion? I'll leave it to your discretion what to skip. So
all right.
Usually what the guideline is, you at least take the first part and then the last part.
Right.
So I think we could just start with the first part, and maybe we just go to the conclusion, and then we can skip around. How about we do that, like the first
Okay.
page and last page?
All right. So Sternberg writes a little bit about himself and his qualifications. They're extensive. I'm not going to read them all, but they're extensive.
If you want to post it up here, you can.
Yeah,
brother.
let me share this screen here real quick, and then I'll also drop this link in the chat for anybody who wants to go read this for later. Come on, where is it? There it is. All right, I'm going to drop this link in the chat as well for anyone who wants to read that for later. All right. Sternberg. Now, again, his qualifications are extensive. I'm not going to read all the qualifications because if I do that, I'll be here for 10 minutes. I began my university education in the early 80s as a committed Darwinian undergraduate who was a strong opponent of young earth creationism. As a teenager, I was repelled by the strain of fundamentalist Christianity that surrounded me in the Deep South, the anti-intellectualism, the cultural flatness, and the pessimistic fatalism that seemed to go with the former two. Equally revolting to me was the accommodationism that I saw in the post-Vatican II Roman Catholic Church that usually took the form of liberation theology on the one hand or bourgeois moralism on the other. Like some of my peers, I was actively searching for an integrated picture of the world, and I just could not find it in what passed for Christianity. Then 3 eye-opening events occurred that led me to Darwinism and immediately thereafter to implicit atheism. For one thing, I read Darwin's Origin of Species and found myself convinced, or mostly convinced, that the author had made his case. For another, I started meditating on the works of Friedrich Nietzsche. Oh, that's great. His writings appeared prophetic, for not only had he accurately diagnosed, in my opinion, the disease, the, quote, last man syndrome, you could call it, whose societal symptoms were and are everywhere to be seen, but he had also foreseen the major intellectual trends of the 20th century. And then I ran into Richard Dawkins' The Selfish Gene and mentally devoured it. By the age of 20, I was an intellectually fulfilled atheist like Dawkins. Having had a lifelong interest in all things biological, my childhood goal was to become an ichthyologist. I decided to pursue a bachelor's degree in biological sciences at the University of South Carolina. As an undergraduate, I took as many courses having to do with evolutionary theory as I possibly could. And it was there that I developed to an art and a most dangerous habit. I would spend hours in the library reading. Not just required materials, mind you, but heretical volumes. And not just your run-of-the-mill books that presented some crankish ideas, but the strong, plain brown wrapper stuff. Literature that posed hardcore, sophisticated challenges to the Darwinism I had so casually imbibed. Day after day, and sometimes night after night, when I wasn't out and about, my attention turned to Richard Goldschmidt's The Material Basis of Evolution and Theoretical Genetics, Hugo de Vries, The Mutation Theory, and Species and Varieties and Their Origin by Mutation. Oh boy, Soren Luftrup. Oh boy, I'm sorry, guys. I'm sorry. I've even cited that person. I still don't know how to pronounce their name. Epigenetics, a treatise on the theoretical biology and Darwinism. The Refutation of a Myth, to name only a handful. None of the authors were creationists, and none, to my knowledge, ever mentioned the G word. But all were evolutionists who were critical of Darwinism and, given their backgrounds in genetics and embryology, able to outline in detail why the facts that I knew to be true were either misinterpreted or erroneous. And then he goes on and digs into a bunch of this stuff. I'm going to kind of skip down a little bit because there's interesting stuff here, but most of you are not going to nerd out about it like the rest of us. Let's see here. Come down to the end, I guess, and then we'll skip around a little bit in the middle if there's anything interesting. Because this is, again, it's a 12-page, I guess, testimony. I don't know what you'd call it. All right, so
you might want to go earlier than the biologic, maybe the Smithsonian controversy.
Yeah, that's going to be. Where is this? Where is the? I'm trying to find this. I apologize, because I have not read this before, but
neither have I.
Okay, so that's apparently.
Oh, there it is.
Yeah, it's going to be up here.
Congressional. Yeah.
All right, so here we go. Then in 2004, I was contacted by Steve Meyer, who told me that he had a manuscript on the Cambrian explosion and the origin of the higher taxonomic categories of metazoans. The central point of the paper, as I recall, he told me, was the informational basis of distinctions among the phyla. He was considering submitting it to the proceedings, he said, whereupon I said that in order to do so, he would have to be a member of the society. Soon thereafter, or perhaps a few weeks, I received copies of the manuscript, and I noticed that Meyer had paid his 2004 membership dues. And I read the manuscript. In the draft I had in hand, Meyer surveyed discussions among evolutionary and theoretical biologists regarding the origination of organismal form, with special attention to neo-Darwinism and self-organization theory, although structuralism was touched upon. I found the paper interesting because here was an intelligent design proponent trying to make his case to a scientific audience. After mulling over the situation, I decided to send it out for peer review. By coincidence, I had to take a mandatory course at the NIH around the same time on the ethics of peer review with respect to scientific manuscripts and grant applications. An emphasis of the course was that it was unethical to shelve papers or applications based on political affiliations of authors, personal disagreements, or because an author/applicant's views are at conflict with one's own. Moreover, the course made it transparent that you do not send manuscripts or grant applications either to those you expect will be presumptively hostile, due to a conflict of interest maybe, or to those who will give it pass without reflection. As managing editor, I regarded the Meyer work as within my sphere of knowledge, and I was interested to see whether it would stand up to scrutiny. So I asked 3 biologists and a geologist to review the manuscript, all of whom I knew to be neither young Earth creationists, nor neo-Darwinians, nor affiliated with the Discovery Institute, nor particularly sympathetic to the intelligent design position. So in other words, he picked as neutral of an audience as he could. I gave each the standard time allotted for such reviews that was specified in the letter accompanying the manuscript. 3 reviewers responded and were willing to review the paper. All were experts in relevant aspects of evolutionary and molecular biology and held full-time faculty positions in major research institutions. One at an Ivy League university, another at a major North American public university, and the third at a well-known overseas research institute. After I collected all the responses to the Meyer manuscript, I paid close attention to the comments. The 3 agreed that the work was original and would generate interest. None found a compelling, non-ideological reason for rejecting the paper, although in their own ways made explicit their thought that Meyer's move to intelligent design at the end would be very controversial. Beyond that, the 3 reviewers had distinct criticisms that each felt had to be addressed. So I sent the manuscript and comments back to Meyer. I thought Meyer could say forget it and look elsewhere, in which case the whole review process would have been an illuminating exercise. Or he could make the changes. The changes might be found to be adequate, and I would have to make a decision. After some weeks, I received a revised manuscript that point by point answered the critical comments, in some cases making additions to the paper. Finally, I had a completed file before me, and thus I could either accept or reject the paper. Do you want to say something, Sal?
No.
Okay. I decided to accept the Meyer article for publication. Personally and professionally, I had nothing to gain from the appearance of the paper. I was not associated with the Discovery Institute. I had resigned from being editor of the proceedings back in October 2003 and only wanted to get back to research. Nor did it seem that I had much to lose. My thinking at the time was that at best the paper would generate discussion and possibly even be followed up by a contra Meyer article, maybe a genteel debate against the backdrop of the question of emergence of taxonomic units. The worst that could happen, I naively thought, was that I would be scolded for having published a work that some might think ventured into philosophy. And to give you a measure of my miscalculations, I seriously believed that the article would be mostly ignored.
I'm sorry. Sorry, go on.
Dr. Sternberg, if you're listening, man, you're naive. But it's understandable because you haven't had to fight this battle your entire life. This was his first real battle. Step into this battle at all. He's not going to be used to what comes next. Scientists tend to ignore works they don't approve of, and besides, the venue was the proceedings, not Science or Nature. So in other words, it's not a high-profile journal. Needless to say, my predicted outcomes were incredibly, unbelievably wrong. I won't go into the details because many of the events that transpired are now a matter of public record. What I will say, however, is that rarely did anyone attack me or the Meyer article on the basis of its actual content. In fact, those who did read the paper and who did object to Meyer's case for design at the end of it tended to be civil in their correspondence. No, the 2 kinds of angry responses that I invariably received invariably began with a statement about how, quote, the offended party did not or refused to read such, quote, pseudoscientific or, quote, creationist nonsense, followed either by a denunciation of my assumed motives or how the paper was a tool of some vast right-wing conspiracy to which I supposedly belonged. And when the situation at the museum became really nasty, I could have used a stopwatch to mark the seconds from the start of a conversation about the Meyer article to tirades about Christians, fundies, Republicans, George Bush, etc. that so often ensued. Most of these foot-stomping, shouting-down episodes I chose to avoid. For if I did not avoid them, I would then hear a rant about abortion rights and the need for stem cell research, and how we must leave Iraq now, and how the Bible is the most dangerous book ever written, and how the U.S. is, was, and ever shall be the rightful home only for progressives. Folks, you think evolutionary biology is not political and religious? Case. 3 things I could not abide, though. First, an act of legerdemain occurred through the cooperation of the Biological Society of Washington and the National Center for Science Education. Oh, the National Center for Selling Evolution. Okay.
Yeah.
The NCSE supplied talking points to the BSW leadership and pushed the BSW to issue a statement that implied editorial malfeasance. Once the BSW did so, the NCSE then pointed to the statement as independent confirmation of wrongdoing, a statement that they basically wrote.
That's pretty slimy.
That's standard political operation right there. That's pretty standard. Second, government officials on government time using government computers were emailing to the world what were at best libelous rumors and often invented tales. My attempts to provide documentation supporting my side of the situation were suppressed. Third, after a failed attempt to have me fired from the NIH, which would have succeeded had Capitol Hill not intervened, the chair of the invertebrate zoology department in the museum told me not just that I was on the wrong side of the political spectrum and thus a threat to many, but that if anything went wrong in the museum, a manuscript missing, a purse lost, or stolen, anything awry, I was going to be blamed. So in other words, evidence doesn't matter. Kind of like how evolutionary biology always works. My research was severely curtailed and placed under the supervision of an opponent who was given complete control over what I could and could not do, and what I could and could not write and publish. My keys were demanded from me, and I was ordered not to go back into my office, which permitted the museum to then blame me for not coming into my office on a regular basis and adding alcohol to the specimen jars that remained in there. I could go on and on. It was surreal, like a David Lynch adaptation of a Kafka novel. With open hunting season having been declared, I presented my case to the U.S. Office of Special Counsel, OSC. OSC attorneys found evidence to corroborate my claims of retaliation and harassment and concluded that, quote, it is clear that a hostile work environment was created with the ultimate goal of forcing, unquote, me out of the Smithsonian. But the OSC could not proceed beyond its initial investigation because it determined that it lacked jurisdiction. Later, a broader investigation was launched by congressional subcommittee staff, which resulted in a finding that there was, quote, compelling evidence that my, quote, civil and constitutional rights were violated by Smithsonian officials. So, folks, if you think that like the stuff that happened over the last 4 years was bad, this was happening in the early 2000s.
Yeah, yeah.
Right.
There's persecution.
Mm-hmm.
I mean, people say creationists are not ejected out of the academy or workplaces. It's like Richard Sternberg wasn't even a creationist.
Yep.
He was just kind of guilt by association.
Yep.
He let one phrase go through: intelligent design in a peer-reviewed publication. If you want to, I think the last, we could, it is a 12-page document. Maybe we've at least given the viewers a lot. I'd like to see the last part.
All right, we can keep reading. We can keep reading.
Yeah. By the way, he's a very good writer.
Yeah.
Excellent. So I'll leave it to your discretion, Emory. Go ahead.
Figure out where I can come up with this.
Now he's explaining how he ended up with the Discovery Institute. And
Yeah, so I'm
we
just
don't
going to
have-
read the last little bit here, the last paragraph,
Yeah.
I think, last couple paragraphs. This past year, I decided to become a research fellow with the Biologic Institute in Seattle, made possible by a research fellowship from the Discovery Institute. Certain Darwinist detractors will no doubt shout, "I told you so!" upon hearing news of this, since some of them alleged falsely that I was a closet ideer working for the Discovery Institute at the time the Meyer paper was published in 2004. The truth is that I only received a research fellowship from Discovery to work at Biologic after I resigned from the NIH in 2007. I'd grown profoundly tired of intellectually swimming upstream at both the Smithsonian and the NIH. Condemnation of my past, quote, indiscretions, coupled with attempts to pigeonhole me in existing creationist evolutionist categories, not to mention the refusal by many to hear me out, sharply circumscribed my freedom of intellectual and scientific inquiry. I was tired of being scrutinized and monitored and questioned and blamed. I was tired of being told that my structuralism reduced to creationism by those who have no understanding of either. By the way, my taxonomy peers at the NIH did not behave this way, and at least from my perspective, we all got along splendidly. Hence, I made the decision to leave Washington, D.C., to move to Florida, to think my thoughts in peace, and try to get back into research. But then the possibility of working at Biologic presented itself last year. Now, the foremost question I raised before accepting the position concerned my intellectual freedom. Would I have greater freedom at the Biologic Institute to pursue the scientific evidence wherever I thought it led? Or would I face a whole new series of litmus tests, this time from intelligent design proponents rather than doctrinaire Darwinists? I was assured that no such limitations would be imposed. My next query turned to the kind of research that would be asked of me, and I was assured that would be left up to me. After weighing the various options, I said yes, and those promises have been kept. My current research focuses on increasing our understanding of the causal relationship between the genotype and phenotype, or DNA specifications on one hand and morphological ground pattern on the other. Specific research projects include examining so-called non-coding chromosomal sequences for the codes that they contain and the ontogenic information that they bear. Most and perhaps all DNA in complex genomes, such as those of mammals, are transcribed, although only a small fraction of these specify proteins. The complex organization and controlled expression of these non-translated RNA encoding elements suggests that an array of encrypted texts awaits further discovery.
And he did, in other parts, gave an eloquent defense of intelligent design, even though he didn't embrace it. He's a very nuanced thinker.
Yeah, he doesn't seem to think in the usual categories that everybody else
No, not at all. And I have to say, I feel a lot of kinship to him, because sometimes when I was in Christian circles and creationist circles, they're very doctrinarian. I said, you guys may be right. It's the method. Sometimes I don't like it being shoved down my throat. That I was grudgingly a young Earth creationist until I went to grad school, because they're saying, oh, the distant starlight problem's not a big deal. I just like, no. And it's when I went to grad school, I began to see that, oh yeah, okay, now I can start to doubt the Big Bang. It was when I saw it for myself, rather than just affirming what everyone else said. That's just my nature. I don't mean to be kind of rebellious like that. But what they say is that these passive aggressives, they're real nice. And someone will say, we have to deal with a problem here. The passive aggressives say, oh, there's no problem here. That's kind of like me. It's like, I'll just mingle with the creationists, and unless they, if they confront me, it's like, well, there's no problem here. I just want to, let me go off and research this. I may disagree with them, but I don't want to be, I'm not like Maddox, who loves getting in people's faces.
Maddox, God bless you. I can't do what you do, man.
But I mean, I will confront the ideas in my heart. And I'm like, a lot of times I will not just articulate it, because I could be wrong in my doubts, in my skepticism. And I said, there's no reason to make enemies. This is kind of my policy. Don't make enemies just for fun, or just, don't hurt people any more than you have to. And so sometimes I'll think, I'm just going to hold my tongue. I disagree, but I'm just going to hold my tongue. We'll get along and build a working relationship. Maybe next year I'll come back and say, hey, great job on your presentation, when I'm convinced that someone's right. And I have done the opposite too. Later I'll say, I think your ideas are terrible. I'll try to be a little bit more diplomatic than that though. So in reading Sternberg, I said I could feel so much kinship with his thought process. Definitely an out-of-the-box thinker, and very brilliant and dedicated. When he was talking about how he's just spending time in the libraries reading.
I can sort of relate to this, except I did my reading in my room rather than the library. But
well, that's the internet age now,
Well, even when I was in college, we still did books. We still did a lot of books, like because I started in 2009, so we were still doing books a lot of the time. But yeah, I did most of my reading in my room or at my job, because a lot of the times I worked residence hall desk when I was working in college, and when I was doing that, I could just sit there with a book, man. I'd sit there with a book and read my book, and look up every once in a while when somebody came in the door. It's like.
Well, it's going to be interesting, and you and I come from different backgrounds. I did not come from a young Earth. Did you come from a young Earth background, Emory?
Yeah,
I did not.
I did.
And when
it was not something that was highly emphasized when I was a kid.
Yeah. But Sternberg, he said something that resonated with me, was the doctrinarian attitude. He did not like that in the South, and I did. It's kind of hard because I don't. It's been a conflict with me and other young Earth creationists. I'm the type, you have to kind of persuade me first. You can't just, sometimes because when I nearly left the faith, there was a lot of shunning and shaming, and I'm like, that's just not the way to build my faith. Okay, it was really hard to deal with. It's like, I'm willing to believe you got to give me some persuasive arguments, and I'm still that way, and it put me at variance with a lot of young Earth creationists. That's why I felt more comfortable in the ID community. They're very welcoming of like, okay, you're a Richard Sternberg type, but if you know your stuff, we love you. You're a free thinker. You're skeptical of both sides. Hey, that's cool.
Yeah, the ID community.
I'm sorry.
The ID community basically doesn't care what you believe, so long as it's some vague form of "I'm open to ID," they'll work with you.
Yeah, and if you're qualified, Sternberg was obviously qualified. He had a fabulous mind. So that's one reason I have a lot of strong ties to the ID movement. And by the way, I'm going to be at the ID conference hosted by the Discovery Institute in Dallas, January 22nd, 2022, this year. So in a couple of weeks, I'll be down there. Anyone who wants to do a meet and greet, we can do that. Although there'll be so many much more cooler people there than me, but I'd be happy to meet you all. I think the early bird registration might expire today. So otherwise you have to pay $45 instead of $35. Just thought I'd let you guys know that. So, but I think I'll just say this, I thank the Lord that I'm alive in this era. I think if I had been born 100 years ago, I don't know that I'd be a Christian. So that was God's mercy to me, that given my temperament, I'm in an era where the evidence is available to me, things that would be persuasive to me of the creationist, the young cosmos, young Earth creationist viewpoint. Because it's kind of scary to think where I would be not for the grace of God on my life. So you could tell I'm a big Richard Sternberg fan. Anyway, thank you for the reading, Emory.
Sure.
Great job.
Sure.
So now let's look at the next. Sandy says it's no coincidence, Sal. Guys, this show is so compelling that I'm missing a lot of the comments. I'm just focusing on the material that we're reading because it is pretty intellectually thick. So let me see what's next on the docket here. And okay, we can just read some abstracts here.
Okay.
On molecular clocking. And reading the entire paper is not going to be of any benefit to me personally because it's in microbiology, which is not my specialty. So people often confuse molecular biology with microbiology. Can you explain the distinction?
Sure. So microbiology is focused on organisms that are too small to be seen with the naked eye. And some very few organisms that you can see with the naked eye, but you really see a lot better under a microscope. That's the focus of microbiology. Molecular biology, on the other hand, is more focused on things like mitochondria, enzymes, proteins, that sort of thing. That's more the focus of what you get into with molecular.
And the way that I understand it, yeah, the way that I understand it, and the way that Change Tan teaches it, molecular biology centers around the central dogma of molecular biology, which is the translation of DNA, the transcription of can't even do this right. Transcription of DNA to RNA, RNA to proteins, and protein. It's kind of that one-way direction from the DNA to proteins. You can't really go back. RNAs are kind of funny. You can have the reverse transcriptase and put the et cetera, some little corner exceptions. So that's also one way to describe molecular biology. It focuses on that. Sometimes it goes a little bit beyond that. So somewhere around there is lots of biochemistry. And then the cellular biologists study the architecture of the cell. And we have all these other specialties. So what I was saying, this is a microbiology specialty. It's not, there are probably lots of things I'm missing here and I won't do justice to it, but the moral of the story is they had to revise, I'll just read it, but they had to revise the affixed date from a very long time ago to something more recent. And I found several papers like that. And you could see why this would be of interest to young Earth creationists. So this time, I'll try to do the reading. And thank you, Emory, for helping me out with
no problem.
With Richard Sternberg stuff.
No problem.
Let me see if I can make this a little bigger. This is... Boy, I hope I get published in PLOS someday. We're trying. Cholera caused by Vibrio cholerae erupted globally from South Asia in 7 pandemics, but there were also local outbreaks between the 6th, which is 1899 to 1923, and the 7th, 1961 to the present pandemics. All the above are serotype 01, whereas environmental and invertebrate isolates are antigenically diverse. Okay, even in the first 2 sentences, this is way out of my league. And plus, it's talking about historical things. And this is one thing you'll see a lot now in biology. Everyone has their specialty. You have the cholera specialty. Now you have the COVID specialist. So everyone in microbiology is also really specialized. The pre-7th pandemic isolates mentioned above and other minor pathogenic clones are related to the 7th pandemic clone, while the 6th pandemic clone is the same lineage but more distantly related. And non-pathogenic isolates show no clonal structure. Okay, guys, there is a point to me reading all this, all right? Just bear with me. To understand the origins and relationships of the pandemic clones, we sequence the genomes of 1,937 pandemic strain and the 6th pandemic isolate and compare them with the published 7th pandemic genome. Oh my goodness, they're really talking to a specialized audience. This is just like so specialized. Okay, We distinguish mutational and recombinational events and allocated these and other events to specific branches in the evolutionary tree. There are more mutational than recombinational events, but more genes, and 44 times more base pairs changed by recombination. We use mutational single nucleotide polymorphisms and known isolation dates of the pre-pandemic and 7th pandemic isolates to estimate the mutation rate, and found it to be 100-fold higher. Found it to be 100-fold higher than usually assumed. We then used this estimate to the divergence date of the 6th and 7th pandemic clones to be about 1880. While there is a large margin of error, this is far more realistic than the 10 to 50,000 years ago estimate using the usual assumptions. We conclude that the 2 pandemic clones gained pandemic potential independently, and overall there were 29 insertions or deletions of 1 or more genes. There were also substantial changes in the major integron attributed to gain of individual cassettes, including copying from within or loss of blocks of cassettes. The approaches used opened up new avenues for analyzing the origin and history of other important pathogens. Oh my goodness, that was painful. Okay, so we're talking about life as a graduate, creationist graduate student. There it is.
Keep in mind, folks, the abstract is the least technical section of the paper.
And I have to tell you, sometimes the professor, when I was in physics class, I'll never forget, he gave me a paper, he gave a homework assignment. He said, guys, read the Sachs-Wolf effect. I crawled through it and I went back. I said, Professor, I think I understood 10% of what was said. He said, that's all right. Well done. You get an A for the class. It's not unusual to be just thrown into the fire like that. We have no clue what you're reading. And so creationist researcher is going to go to the labs and, well, not labs, but to the papers like this, trying to dig for that needle in the haystack for a clue. And, okay, what did I get from that? They had to change their estimate of the date by 100-fold. So instead of 50,000, they're bringing it down to 5,000 or something like that. Is that relevant to anything else in young Earth creationism? I would put it as a big maybe at this point. I would need to talk to the microbiologist and say, okay, what does this mean? But, this is why we would have like creation conferences, meet people. Mm-hmm. One of them is already outed himself, Andrew Fabich. He published in Nature, and he was in the Ken Ham, Bill Nye debate. He's the microbiologist that was featured there, and he said, I'm a young Earth creationist, I've been published in Nature, et cetera, et cetera. So I would take this and say, hey Andrew, what does this mean? Is this a change in mutation rates, or do you have some population genetic model? Go on.
So I think I can actually tell you a little bit about what this means.
All right.
Not without reading the paper, but just reading through the abstract. What it sounds like is that they had a PopGen model that explained the divergence of the various clonal strains of cholera. But when they actually checked the mutation rates, what they found was, oh, it's not 10 to 50,000 years ago. It's only about 100 years ago. A little bit more than 100 years ago at this point, probably 120, 130. But it's only about 100 years ago that these two diverged from one another. That's basically, if you boil all that down, that's what they're telling you. Is they're telling you that, hey, these pandemic clones of cholera, the ones that cause disease, they diverged a lot more recently from one another than we thought they did. That's basically what you're getting out of that.
And that's kind of what I was getting to it. And they're pointing out there were insertions. Mm-hmm. And so that can really monkey with how you do this. I think the takeaway from this, and this is what I said, why I loathe genetics, especially trying to reconstruct how far back in time, because of the uncertainties. I mean, this is, for a physicist, this is anathema. It's like,
Mhm.
Physics, you're measuring like Planck's constant to umpteen decimal places, real precision. But this is just like, what do you do with this? There's so many unknowns. And so there's a big debate between ongoing adversarial relationship between Dan Stern Cardinal and Nathaniel Jeanson. Obviously, I'm on the unearthed side, but I'm not really quick to rule on either side, because this is one reason I'm not, even though, and Nathaniel and I have suffered together, persecuted. We were there, Lipscomb University. You remember Dr. McClure, Dr. Jeanson, if you remember that incident. Both of us took bullets together, so this is not meant anything personal to you, Dr. Jeanson, but it's these levels of uncertainty that make me feel uncomfortable. Whereas when we were talking about Richard Sternberg stuff and some of the other stuff, it's like that seems more tangible because we're dealing with how biology works today.
Mm-hmm.
When you're trying to reconstruct historical events from what we have today, that's a tough road. That honestly is. And Stephen Meyer said he argues for ID, not so much kind of the timeline and the chronology. And that is a lot easier. It's easier to say that Stonehenge is built. Much harder to, or the statues at Easter Island are designed. Much harder to say when and how. Much harder to say when and how. Yeah. So, if you have any thoughts while I try to grab the next one.
Well, no, I think that's a good answer, and I think that's again why I think ID should just be the baseline. Like if you're gonna subscribe to some form of Christianity, ID is like the baseline. Like to me, I don't see that there's any reason to go to a evolutionary position because you simply don't need to. Like the science, the Darwin's book was not great when he wrote it. It's an interesting read, but like it wasn't great science when he wrote it, and it got some very negative scientific reviews at the time. But it has aged, and its spawn, evolutionary biology, has aged very poorly over the last 150 years. I guess it's 170 years at this point, but yeah, it's not aged well. So,
okay, I'm not gonna. This is the second paper, and I'm not gonna read. I'm not gonna suffer myself and the viewers through the entire abstract. I'll highlight the part that's big. Mutational synonymous SNPs were used to estimate a divergence time of 400 years using a new clock rate, in contrast to 14,000 to 70,000 years using traditional clock rates. Okay, so that's a big factor, guys. And this reflects kind of the ongoing fight between Nathaniel Jeanson and Dr. Dan. And accusations are floating around, and I'm in the middle of this. And I also I still work with Dr. Sanford, but not as a paid employee. And so you know where my sympathies would naturally lie. But I wasn't hired by Dr. Sanford to be a yes man. And that sometimes caused a little friction because I tell him, I know you want to say this, but I don't think we can justify it given what we know. Some future discovery might affirm that viewpoint. But I'd always say this, word this in our paper a little bit differently. So all this to say, if we're seeing these massive revisions in secular review, it's like, okay, what do we really know? I mean, it's heartening to see that they're revising the dates to more recency. Mm-hmm. That's why this, the paper is of interest to me. So I'm just throwing that out there. And the next paper will also put some wrinkles into this. Now, I will point out that the microbiological world might have a lot of wrinkles in the molecular clocks because microorganisms are different than multicellular creatures like humans and dogs and cattle. But still, the point being, there are a lot of unknowns and I think it's premature to be doing clocking and have, you know, confidence you've got a good historical reconstruction. So that's kind of the moral of that story. If you have any other thoughts, and again, this is so technical, I'm just like, I'm not going to, I'd have to talk to a microbiologist to find out, to uncoil what all this means.
Mm-hmm. The primary interest is the clocks. Now, one thing I found very interesting is in my evolutionary genetics class that I took over the fall semester, they actually surprisingly suggested that the clock data may not be the most reliable and to be very careful when you use them. So I think the Evo community might be waking up to the fact that, you know, yeah, we can use these clocks in the public to, you know, wow the unsuspecting, but behind closed doors, we're not so sure that this works.
Yeah. And you know, as much as I want to tell, this is what I wanted to tell the young Earth creationist community. I said, you know, maybe reassuring you can get one date from the DNA clocks, but I think the geological clocks, the other clocks that people like Russell Humphreys and all are doing, those are more, I think they would be more potentially precise. We just don't have the money right now for like the helium diffusion in zircons, for example. People say that's debunked. I said, you know, the problem, I wanted to say huge timeout. We could always redo the experiments if we had the money, and we could do it in much better controlled conditions and evaluate all the instruments. We could do this if we really wanted, but you know, there's not a lot of incentive to get it right, if you know what I mean. Yep. I see that with the carbon-14 dating, but so my bent is the molecular clocks, the biological clocks. It's like, you know, I would not make that the best argument. I think we can put bounds on the fossil record being like not much more than a few million. I would feel very confident in that. I'm not going to immediately argue. I mean, if we just took a snapshot where we were, if we took mainstream science without any revision to physics, if we accepted the age of the solar system from the mainstream, we could make a very good case the fossils died recently. And that would be based on chemistry and then some other considerations in radiometric dating. I didn't say we'd make an airtight case, but I wouldn't want to be on the other side. Just like when we were kind of talking about Mary Schweitzer and she lost her young Earth creationism. And as you were saying that, and you said, God just handed it to you on a platter there, Mary.
Yep.
I mean, of all people that he would choose, and a Sandy Pigeon would say there are no coincidences. I hope maybe when she retires from all this, she'll ponder and say, you know, maybe the Lord was speaking to me. I really do feel for her because she wasn't, her account was she got saved in college. And I regard that more heavily than someone that was raised from childhood. You know, someone that came from a non-Christian viewpoint in college to become a Christian. That's why I believe she's a Christian. But anyway, now we got some of the more juicy stuff. I'm not going to read the whole thing, but this is, I didn't even notice Lenski was an author of this. Let me show it. Let me show. So this is Proceedings of the National Academy of Sciences. Oh, let me see. Click all author. Oh, Richard E. Lenski. All right. So to get published in the Proceedings of the National Academy of Sciences, you need to be, it's not unusual for the National Academy member to put his name in there, and that paper will be automatically published because I guess they regard you so highly that you could say whatever you want, which is kind of cool. And you can basically come into a peer-reviewed journal unless it's just so outrageously stupid. Then they might pressure you to make a retraction. There were some There was one paper like that by a biologist. It was really kind of funny. I don't remember which one now. Otherwise, you would have to approach them if you're an outsider, and he won't sign his name to it, and they'll just say this paper went through so and so. So good on you, Emory, to have found that Lenski was co-author. I didn't even notice that the first reading, but I'll read the abstract and conclusion. It's very telling. So I'll read the significance. Bacterial genomes are extremely diverse in size and composition. Biologists have long sought to explain such variability based on present-day selective and mutational forces. However, mutation rates can change dramatically over time. Oh, not good for the molecular clock. However, mutation rates can change dramatically over time. And experiments with hypermutable bacteria show that their genomes rapidly decay when propagated under the near absence of selection. Whether selection can prevent this decay is unclear. Yeah, like no duh. Here, we document the rapid genome decay of hypermutable bacteria even during tens of thousands of generations of sustained adaptation to a laboratory environment. These findings suggest the need to re-examine current ideas about the evolution of bacterial genomes, and they have implications for other hypermutable systems such as viruses and cancer cells. And
congratulations, Dr. Lenski. You've just found genetic entropy.
Yes!
I don't think he would like that if he heard that, but that's what he's done.
Yeah. So they go over his own experiments, and I think they were a little bit gentle because he was a co-author. I would have ripped into him. They should have had Michael Behe as a co-author, I think, personally. You would have gotten a much more accurate characterization. So they keep mentioning here LTEE, also all over the place. This is Lenski's experiment.
Mm-hmm. Yeah, and
how does Scott through? Yeah,
Lenski's,
go on.
While Lenski's experiment, the way it gets spun is, atrocious. I agree with Dr. Galuza. I want this experiment to go on till the end of time because I think his populations are all going to crash eventually, and then we'll just get to all sit there and go, "See, we told you so."
I think. Well, we do know some of the lineages died.
Yeah.
So, okay. So this is really good. They're going. Oh my goodness, this is more detailed. I just wish they had a more forceful conclusion and said, all you said, Dr. Lenski, is absolute bunk. And you're misrepresenting it to the public. Shame on you. But of course, they won't do that. Oh, this is a miserably long paper.
In other words, I get to read it tomorrow. Yay.
All right. So, wait. So this is the materials and methods. I'm sorry. Let me get to. So some places put the materials and methods after the conclusion. Let me see if I could find it. I'm going to have to use the search tool here. Hang on.
You're going to want to go to the discussion section, I think, because that's that's before the materials and methods.
Now, I really want to point out something that says they use the word decay a few times. So, like in this example, it says, second, the fastest genome decay rates are observed in the LTEE amount to 1% change in GC content. So we see the word decay. This observation suggests that draft-induced decay might occur even in non-mutator asexual populations, but such decay might be prevented in relevant timescales too long compared to just blah, blah, blah. I'm sorry, blah, blah, blah. Let me get to the... Okay, I think we're getting to the conclusion here. Okay, finally, our results also have certain implications for biotechnologies. Researchers working on biotechnological applications may be tempted to use mutator strains, that is Lenski's type bacteria, or chemical mutagenesis to accelerate strain improvement. Consistent with this strategy, with that strategy. The LTEE populations that evolved mutator phenotypes have reached slightly higher fitness. They've reached higher fitness levels
While their
than
genome's
those
falling apart.
that did not. However, the increased mutability also comes with potential costs, as shown in this study by the tendency toward genomic decay. In earlier studies by reduced performance under environmental conditions, under other environmental conditions. Therefore, unless one seeks to optimize performance under very constant and precisely controlled conditions, the accelerated rate of adaptation might not be worth the resulting genomic decay and associated loss of versatility. And I got to tell you, Emo, he came on this channel and was just pumping out drivel, and I told him this is junk, garbage, LTEE. And several guys came in the comment section and did that. And I kept telling this is a disaster. And I just didn't have time to kind of spoon feed them because they would just regurgitate. He said, look at this improvement. You got it here in a peer-reviewed paper by Lenski himself saying it's decay. And
yeah, the decay might make you better off in a very specialized environment, but it's still decay.
Yeah. So I tossed these guys out of the comment section, and then, you know, accusations of me being a coward. I'm not.
Yeah.
I'm not. You know, I just don't waste time on people that don't want to learn.
Howler monkeys.
Howler monkeys.
Yeah.
You know, there you have it, guys. If you bother to try to read the literature as we have, you might learn something instead of regurgitating talking points.
Yeah. Yeah.
I don't have time to deal with you numbskulls, seriously. And I don't want your junk on this channel putting doubts in people's minds because of the falsehoods you're spewing. I don't give platforms to nonsense like that when I've rebutted it several places in this channel, in other things I've written, and more importantly, it's right there in the literature. You don't want to read it because you have your talking points. You don't, you know, sometimes I get mad at you guys, honestly.
Suggestion
you
for
guys
any
think you're so superior. You don't even read your own literature. Sorry, Emory, go ahead.
Suggestion for any evolutionists who decide they want to read the peer-reviewed literature: don't read it to try to refute creationists. Read it to try to educate yourself on the topic, and then once you have done that, if you still think you can refute what we have to say, which I doubt you will be able to do that, but if you still think, then you come back and you say, okay, I've read this paper, and this paper, and this paper, and they say these, this, and this. We can have a discussion. Don't just come at us with talking points. We've heard the talking points before. You're not changing our minds because with those talking points. You bring us the literature. You actually go do your homework like we do it.
Yeah.
You think it's easy to sit down and read through this giant monster that Sal just showed you? No, it's not. But we do it because we actually want to understand what you believe. Show us the same courtesy.
Yeah. And I'll tell you, occasionally, I don't watch the videos that are made about me, but I do see the titles. One of them said, Cowardly Cordova. I'm just like, okay. I know where this probably, I suspect where it came from because I was tossing these guys. All right. Let me give you an example. Dave Farina. He's beaten to a pulp by James Tour. James Tour schooled him. And the guy still keeps repeating, Uri Miller, Uri Miller. I'm just like, RNA world. All right. I mean, you just got taken apart. And this is what I have to deal with. After it's been explained to you that this doesn't work based on textbook chemistry, textbook understanding, you still don't get it. And you're going to come here and heckle and try to disrupt this community with your garbage. I'm not going to tolerate it. If Christians want to learn this, they can go and look it up themselves. I invite them. But they're, I built this place with the expectation they can come here and get to hear our side of the argument clear and unencumbered, because there are plenty of places in the world where they could hear the other side. There are very few places you'll get this level of quality to hear it this way. You'll get to see, we'll get to present you papers that are even in evolutionary literature. How many creationist materials have you read so far, honestly, tonight?
Maybe, I mean, if you're really, really, really stretching the definition of creationist, maybe Sternberg.
Yeah, maybe Sternberg, who doesn't declare him as, okay.
And probably an atheist. So that's a weird form of creationist right there. But anyway.
Yeah, I'm not sure he's an atheist, but that's neither here nor there. This is what I'm trying to say. I'm trying to tell the Christians here, if you're a Christian, you're a creationist, and you're struggling, you're biology, you're or other science student, you can talk to me and say, hey, my professor gave me this, and it really bothers me. Mm-hmm. And I'll talk to you. You're welcome to put that in your comment section, in the comment section here, and say, this bothers me. But if you come here and say, Sal, you don't understand how evolution works, you don't get this or that, and then I tell you to read the papers that I wrote that were peer-reviewed, put in by a respectable publisher in Springer Nature, and you say, oh, it's just a chapter and it hasn't had any citations. You don't respect what I'm saying. You respect more that people just pat themselves on the back in the evolutionary community. You didn't give me the courtesy, and you're going to try to educate me. And I could tell you my co-author, he had 14 PhDs under him in Ola Hosjer. Probably similar story, John Sanford and Bill Basner, Professor Emeritus in mathematics. And you're going to say, I don't understand. Ola Hosjer is a professor of population genetics, an award-winning mathematician. If I said garbage, he would not put his name beside me as co-author. I was very honored. Yep. Instead, you just have this drivel that I don't understand what I'm talking about. That may well be the truth. But guys like you haven't proven that you know your stuff.
And if it is true that, myself or Sal makes a statement that's not true, fine. We'll accept correction. Show us.
Yeah.
Bring the papers. Show us where we've said something that is not backed, supported by what's in the literature. Like, I get that you're going to say, oh, creation is in the literature. Duh, we know that. But when it comes to just the pure science that we talk about, bring the papers. Well, I want to see them. And I've gotten to the point now where when I get a troll on my channel, I will just ask them, okay, where's this in the literature? Show me in the literature. And that usually ends the conversation because they don't know where it is in the literature. They're just repeating talking points that they got from Dawkins or Dillahunty or some other.
Aaron Raw. I'm sorry.
Aaron Raw. Oh, please. But they're just repeating talking points. They haven't actually educated themselves on what is in the literature. I used to take some of these guys seriously. Like when they'd show up, I'd actually go, okay, well, maybe I'll consider that. I don't anymore because every time I've done that, I've gone, oh yeah, you have no clue what you're talking about. So I just kind of go, okay, show me the literature. Go, you show me the literature. And this is why I'm willing to dialogue with someone like Andrew Cumming. I know there are certain people out there who are not, but I'm willing to sit down and have a chat with him because Andrew will at least pull out literature, pull out papers and say, okay, here's what this says in this paper. Here's what this says in this paper. We talk back and forth, toss papers at each other. That's fine. I'm down for that. But this whole, oh, you don't understand evolution because drop dead. Just go away. If you want to actually have a conversation and we can talk about the literature, fine. We can do that. Don't come to me and tell me I don't know what I'm talking about. I guarantee you I've read more evolutionary papers than you, unless you're Dr. Dan. Right? But then Dr. Dan is welcome to come have a chat with me because I actually know we can have a conversation and not kill one another.
Yeah. And by the way, Dr. Dan gave us the all clear to advertise. We're going to be on a Ask Me Anything with him. And I've been debating whether I would reciprocate and do an Ask Me Anything, Ask Sal Anything, because I've been banning trolls and howler monkeys off of this channel. I'll give them one shot at me. And we'll just see how that goes. I haven't decided if I'm going to do it. But let me bring this up again, because it actually highlights the peer-reviewed chapter that we published. And I don't think it, Dr. Dan gave some of his criticisms, but central point, evolutionary fitness is not the same as functionality. And ironically, this paper kind of like, oh, it says here, you see where it says this. It's talking about the increased, it says higher fitness levels, but then there's decay. And that's what I was pointing out. In the evolutionary definition of improvement, sometimes they'll conflate reproductive success with improvement. This is one of the first papers that I've seen where they are very clear to make a distinction between the evolutionary definition of fitness and genomic decay. Because it always used to be that more reproductive success implies genomic improvement. Because in some of Michael Lynch's paper, they'll talk about compensatory mutations. Compensatory mutations, when it's the genome's getting trashed, you get these compensatory mutations that increase fitness. And that kind of bothered me. Is that meaning it's recovering function? This paper shows that no, it just means it can make slightly more babies. And so how would you characterize what genomic decay is?
All right, so genomic decay basically is the breakdown of the genome over time. So the genome undergoes a process we call genetic entropy. There's other names for it in the literature. They call it other things.
Reductive evolution is one.
Reductive evolution.
It's not the most comprehensive term. But this is the first time I saw genomic decay.
Genomic decay.
If I wrote a book that said genomic decay instead of genetic entropy, it's like, well, okay, that wasn't a creationist word. I'll go on, Emory.
You're right. But there's also I've also seen Muller's ratchet for asexuals, where basically what happens is you end up with a buildup of you get so many mutations per organism per generation. Now, some of those do get selected out, right? Some of those, the real bad ones, that selection gets rid of those and says, nope, we're not getting rid of that. But a lot of them don't. They just sit, they're either neutral or nearly neutral. They don't really cause a major change, or they don't really cause major functional damage. And so selection's blind to them. Selection can't see them. And so they build up. And as you build up over time, this just, it's like, think about it like a guy walking down the road, and every time he passes a stop sign, which would be a generation change, you add an extra backpack on him. So, when he first starts walking down the road, he's only got one backpack on. Past the stop sign, now he's got 2 backpacks, then 3, 5, 15. He's going to start slowing down over time, right? Because he just can't drag all that stuff that he's carrying, right? And by the time he gets to stop sign number 20, he's probably crawling if he's moving at all, right? Well, that's what genomic degradation is. You just get this added baggage that the genome has to carry around, and it builds up and it builds up and it builds up and it builds up. And eventually it gets to a point where it will literally mutate the organism right out of existence. And that's what happened, by the way, to the, I believe it's the influenza? Oh, goodness. Rob Carter talks about this, where they had the flu. It was the flu. Where the old flu virus has mutated itself out of existence. The human flu, the one that's currently circulating in humans, is the swine flu, because the old human flu mutated itself into oblivion. Go ahead.
So there is some subtlety when they say mutational load. They often still describe it in terms of Darwinian in reproductive success.
Yes,
what we call W, capital W for absolute fitness, lowercase w for relative. But this is the first time they're making a distinction so clearly. That fitness is not the same as functionality. And so how do I define genomic decay? And I'm starting to be behind in my audio. I may have to go to my other computer. let me see. I'm gonna hang. Can you hang on a bit?
Sure. Yeah, so I guess Sal's swapping computers at the moment, so we will go. I'll just go ahead and talk. Hopefully, the analogy that I just tossed up there for you makes sense. Yes, I'll switch computers. Hopefully, the analogy that I just tossed up there makes some sense. Now, there is some mechanism to reduce this load, where you can go through what's called a purge, and sometimes this happens through a bottleneck event. But it really is dependent on how big the population is, how severe the bottleneck is, and so on, and what species, what gets bottlenecked out, and so on. Now, I will also point out that a purge only might increase fitness, but it does not and cannot remove all the baggage. Like you want to think of a purge as throwing a couple of the backpacks back in the ditch on the side of the road. And the next stop sign he comes to, he has to pick up another backpack. So you can't get rid of all the built up damage. It's irreversible, and there's an extinction clock counting down on every species that exists. Now you can slow that down through outbreeding and through careful breeding with your organisms if you've domesticated them. But there is a clock on species that eventually they're all going to go extinct because of the mutations that they're being saddled with in their genome. So,
well, I'll give also a simpler definition of what I consider genomic decay.
Sure, go ahead.
If the genes and the regulatory regions are just knocked out, as you have more of these knocked out, so when the organism simply starts to lose genes, so notwithstanding what you described, that's the eventual effect. When you start losing all these genes, you're not able to persist. In fact, in a sort of related way, they believe a lot of aging is because the DNA is damaged in the cell, in our cells, in our cells. So I would presume that would mean these genes are being damaged. That's an example of genomic decay. What starts to happen, things just don't work as well. But it's fundamentally because the genes get compromised. There's some mutation in the genes. In the worst case, the gene gets knocked out or equivalent, and it can be knocked out any number of ways.
Yeah,
it could be
some
knocked
organisms
out.
actually will have mechanisms where they will literally knock their own genes out for certain situations. This is something that happens in polyploids, where when you have a polyploid form, sometimes there will be immediate gene knockout on certain genes because of gene dosage and so on. They need the gene dosage on that particular gene to be precise in order for it to work. And so they'll knock out extra copies in order for it to continue to function the way it's supposed to function. So that would be genome decay, even though it's the organism itself is knocking it out. This is also true of parasites, by the way. A lot of parasites have shed significant portions of their genomes because they don't need a lot of stuff because, they're parasites. They're getting their nutrients through something else. But that also would be an example of genome decay. Even though it's beneficial in the short term to the organism, long term for the species, it's bad.
Yes. And in that Lenski paper, which now is gone because I had to reboot the computer.
I've got it open, Sal, so if you want me to share it, I can.
Okay. I don't need it immediately, but it said the word versatility. And that's a very important point. It's just like you have a spare tire in your car. You can actually get rid of the spare tire if you don't get a flat tire. Your car will look just fine. And so there are a lot of systems in biology where you can get rid of a lot of stuff until you're confronted in a new environment, or if you've lost your primary system. So like the space shuttle has five navigation systems. Each of them are capable of navigating the space shuttle back because it's so critical. So you could lose 4 of the navigation pieces and the thing will fly just fine, thank God. So in biology has a lot of this. And what was her name? I forgot. I'm having a Joe Biden moment. I can't remember what. She was a researcher up in Canada, and she was doing single and double gene knockout experiments on yeast. She could knock out like 80, she could knock out so many of the genes and it would work. The fitness would still be there. She said when she did the second knockout, that's when you really saw that the gene was essential, that they were backing up each other. So Brenda Andrews, I think her name is Brenda Andrews.
That name rings a bell. Yep.
And she was advising the ENCODE panel and saying, be really careful about your knockout experiments, because you may think that just because the organism's immediate fitness isn't affected, that it's non-functional. She said that my experiments have shown, even in yeast that are not anywhere near sophisticated as humans, they're redundant at least to one level, sometimes two. In humans, it could be as many as 6 or 7. I don't know. I don't recall, unfortunately, where I got that figure. So in the Lenski paper, they said it loses versatility. So what that means is if bacteria may be, or any organism may be carrying a lot of genes in anticipation of being in another environment than the one they're presently in, which implies foresight, by the way, which evolution is not supposed to have. But, and that really is another evolutionary problem. How do you develop backup systems? Because they're actually, in terms of immediate fitness, they should be selected against. Because they come at a metabolic cost. And we're seeing that with Lenski. What happens if there's when it was under the initial heavy selection pressure, it began dumping, it had to get a mistake, and then that mistake in that environment helped through a series of things. It's like the other ones with even more mistakes that knocked out genes got selected for, and in that environment you started. It's like you got the less capable ones bearing the most offspring. To sort of quote another evolutionist, the ones that got dumber and dumber were the best thing for the population, so to speak. So yes, I'm trying to create a new meme that will go through the creationist community. But versatility, that's the key. That's the first time I saw it, and it's nice that it was all in one paragraph, because that encapsulates the problem of immediate fitness is not necessarily long-term fitness in other environments. So since we're getting up to 2 hours, I'm going to have to save all the stuff on distant starlight for next time, because I'm just going to wear Emory out here, and we were ending on such a happy note. Yep. And let me share something. I'm getting my other computer up. Let me share something. I might, Emory, I might let you go, and I might just have a few words on the physics side of things.
Oh no, you're fine. I can hang out for a little bit. Physics isn't really my cup of tea, but I can at least hang out in case your computer crashes again.
Oh, thank you. That way I could set up the next show that'll go a little bit more in detail.
Sure, that's fine. Yep.
So this is the first time I saw like genetic entropy in an experiment that was highly touted as fitness increasing, and it's the infamous Spiegelman Monster. And it's just amazing. They had the same kind of spin that Lenski was doing with his experiments. Okay, Spiegelman's Monster is the name given to an RNA chain of only 218 nucleotides that is reproduced by the RNA enzyme replicase. By the RNA replication enzyme, RNA-dependent RNA polymerase, also called RNA replicase. It is named after its creator, Saul Spiegelman, of the University of Illinois at Urbana-Champaign, who first described it in 1965. Spiegelman introduced RNA from a simple bacteriophage. Q beta into a solution which obtained Q beta's RNA replicase, some free nucleotides, and some salts. In this environment, the RNA started to be replicated. After a while, Spiegelman took some RNA and moved it to another tube with fresh solution. The process was repeated. Shorter RNA chains were able to be replicated faster. So the RNA became shorter and shorter as selection favored speed. After 74 generations, the original strand with 4,500 nucleotide bases ended up as a dwarf genome of only 218 bases. This short RNA sequence replicated very quickly in these unnatural circumstances. And so, let's see if I got control back. And so this was touted as a great triumph of proving Darwinian evolution, just like Lenski's experiment. If you have some comments here while I try to switch back computers.
Oh, that's fine. No, yeah, I mean, just reading through that, it's just like, yeah, okay, guys, okay, yeah. We go from a massively long genome to 218 base pairs. Not to be rude to the Evos, but like, I'm pretty sure that the DNA, the common DNA experiment that you guys do, where you might have done in the past, where you extract DNA from a strawberry. I'm pretty sure the DNA that you're extracting from that strawberry is longer than 200 base pairs. Like, this is just incredibly. It's incredibly bad to call it. Now, it's interesting science. It's interesting science, but it's incredibly bad to call this evidence for evolution. It's going the wrong way. You're going from a highly complex genome with thousands of base pairs to 200. Right?
Exactly. So sorry for some of the audio problems as I was making the transfer here of control. But this again is an example, an illustration of fitness increase at the expense of genomic decay. But the problem with evolutionary literature, except for papers like this one that I had read that was co-authored by Lenski, that was actually critical of this work, kind of in a maybe not so subtle way. So I do credit him for letting, being a co-author of a paper that's critical of his other work. Mm-hmm. The problem with evolutionary literature, most of the time they conflate fitness with genomic improvement, or at the very least they don't make clear the distinction.
Yep.
And in some of the early, the book Genetic Entropy, it didn't make that distinction as clear. But this was, the book by my boss, Dr. Sanford, was written in 2004. This is before he made his paper in the Quarterly Review of Biology Quarterly Review, where he declared most observed experimental evidence of evolution in the lab has been loss of function. That was a landmark paper. It was one I was waiting for. It's been ignored by the creationist and ID community by and large in their popularized article. And I said, you got to highlight this more, because this is devastating, because it's showing fitness in the evolutionary sense as selecting for genomic decay. And then when Dr. Sanford and I released our most recent chapter, we had a long discussion. I said, John, this is kind of novel. We need to break away from the evolutionary definition of fitness as we describe genetic entropy. If we describe it in terms of functional loss, we're going to have a much stronger case. And, function is kind of like a difficult word to define, but I said one accepted definition of functional loss is just loss of genes. Because the evolutionists will call this reductive evolution. I said, we can go with that. We won't get a lot of pushback. We won't be forced to be defining, what do you mean by function? What do you mean by information? You could say gene loss. Simple enough. That'll be universally understood. Or loss of regulatory, loss of regulatory control. And, you know, so this distinction, I've gotten flack from someone I won't name. This is a good definition of fitness. I'm just like, no, it's not. You have an example now in this paper that I read by Lenski himself. It's like there's a pretty important distinction here, and I'm glad they're using the word decay. They didn't use the word entropy. I'll go with decay. I like that. So if you have any closing thoughts before we get on to the distant starlight problem, and I'll try to.
I think we covered that. I think we covered that pretty well. I think that's pretty much a good wrap on that subject. We move on to something else.
Alrighty. So I didn't have all of my slides put together, and that's just as well because we're running already at 2 hours, and so so I'll cover this in another show, maybe shortly. Maybe I'll give Emory a day off and I'll just do a monologue tomorrow night or something.
Can't be tomorrow night, Sal.
Is it the 10th or 11th we're supposed to?
The 10th. I think tomorrow night is the.
We'll have to look at our calendars.
Yes.
Oh my! I might be on. I'm in trouble now. You know what? You may have saved me, brother. I might be on a YouTube channel with 30,000 subscribers. Oh boy.
That's pretty good.
That'd be fun. Okay. You just may have saved my rear end. Thank you. So well, maybe I'll do that show, and then I'll do my monologue tomorrow night. Do another late nighter. We'll see. So this, just to reassure you guys, this is just going to be an overview, and some of the stuff I'll talk in more detail because this is a tough physics subject. And if people ask, if I had to do like a properly formatted Ask Me Anything, and they'll ask me why I'm a yuck, I'm just like, okay, do you want a 10-hour answer? Because that's what it's going to take. Just to give you a sampling of the arguments. Not going to, no way I could pull this off in 2 minutes to give a short answer. I mean, some people say, well, I'm a yuck because the Bible says so. Well, that's valid if you know, God worked a miracle in your life and you're really persuaded and God's helping you. But for people that are like kind of on the fence, that's not the answer they may want to hear. And I've been there, done that, okay? So I'm not saying that's a bad answer. Praise God for people that honor his word. But that may be a good answer for someone who's persuaded of the Bible being true. Mm-hmm. For someone that's struggling, he's probably looking for another answer. And that's when I said that's when you need the 10-hour version just to get started. So quasars. Quasars, every model does not know what to do with quasars. But they've been problematic for the Big Bang, and they've been problematic for all sorts of cosmologies because people don't know what they are. The reason this is important is it's assumed that because many quasars are strongly redshifted for the cosmology types out there, you know what I mean, that they're strongly redshifted, they must be at the very far edge away from us of the universe, like billions and billions of years away, to quote Carl Sagan. Billions and billions of light years away. And the problem is they may be redshifted. First off, okay, redshifts are real. So when the police radar is trained on you, the speed radar, okay, there is a redshift in that radar signal when you're going away. And you can, relativistic, calculating the Doppler effect, like when you hear the train going at you, it has high frequency. When it passes you, you hear the low frequency. That's a Doppler shift. In general relativity, it's a little more subtle than that. The math is a little bit more complicated, but for lack of a better word, I'll say it's like a Doppler shift. And you can measure that Doppler shift, and you can be confident of that Doppler shift because you have the known frequency of transmission from the police's radar gun. And so when the signal comes back to his radar gun, he can measure the difference between the original signal and the final signal and say it's redshifted, you're either speeding or you're within speed bounds. The problem with declaring redshifts for these stars is you actually don't know what the original frequency was. And I've actually seen some of these plots. YP Varshney was saying, okay, tell me if this is redshifted or not. And my jaw dropped. I said, it's anyone's guess. And there was actually a test given to people to say, okay, is this redshifted or not? He was just throwing up random stuff or some samples. And it's not as clean as you think. I just need to point that out. And in the show, I'll show, he had some, I probably need to download his website because I'm fearing he's really, really elderly. And someone's now running his website. He may be of that age. He's in a nursing home and he's no longer. I tried to contact him. But I'll show you some of those plots. And it's like, oh my goodness, you can't tell whether this is really redshifted or not. You're guessing what the original source signal is. Because if the original source signal had different characteristics, it's going to give you something that might deceive you to think it's redshifted. I need to point that out. So what is it about quasars? We have found that stars can have, they're made of plasma, but then you can have laser action in plasma. And so quasars could be laser stars. No one knows for sure. But if they're laser stars, then the quasar could be a lot closer. What implications does that have for us? The way we're able to judge distance sometimes is what we have. That's why you have two eyes help to give you depth perception. It's parallax. You do a lot of parallax, and we use the quasars as our assumed most distant point. And if it's a lot closer, we're going to have problems. And I know there have been arguments in the astrophysical community about how close or far something is. They said, well, our best says it's this far. And another one said, no, our best says it's this far. This could really blow apart all of our cosmological models to some degree. And we're also hearing other crises in cosmologies. I'm just throwing that out there. That let's not be too quick to say what we see is what we really think it is. So that's one.
So what you're saying, Sal, is that cosmology may well go supernova.
Yes. So, because I've been interested in saying, are things, I mean, just, okay, I'll tell you why I just threw this out, and it's a long shot, and I'll be the first to admit it. But what if the universe's dimensions are not quite what we think? It may alleviate the distant starlight problem a little bit. Okay, it's still not getting us through. I do think some of our parallax measurements up to 30,000 light years are valid, at least. So we still have to account for distant starlight. I'm just saying there's a chance it's not quite as severe as we think. We don't know. Danny Faulkner will get after me for this, and we've had arguments in the back channel on that. And I usually say with cosmologists, just sit and wait. We have more arguments than we have data. It's just not It's just pointless to be arguing at this point. It's not like
some of those cosmologists should come help us in genetics.
Yeah,
we have more data than we have arguments.
Yes. So you sort of imported a cosmologist into the biology realm for 7 or 8 years, and I'm glad I was, because I'm actually productive here versus I've been at the Creationist conferences since 2008, and yet cosmology has not moved forward very far. We're still bickering, whereas you heard James Tour's take down of Dave Farina. That's going to be solid forever. No one's going to bicker over that. That was like glorious. But you don't get that in yet cosmology. So the next thing is the groundbreaking discoveries in the chemical and electrical influence of radioisotopes and radioactive decay. That may be important. When we look at rocks that supposedly have these long ages, if Noah's flood was connected to an electrical disturbance that caused the creation of radioisotopes, or like the daughter products, that's going to be a big deal. It may be that kind of the natural isotopic distribution will look like it's old. And, because someone said, well, look at the moon rocks, it has the same profile. And I said, that's actually a good argument. And then I looked at Walter Brown's work on this as articulated by Brian Nichol, and I was like, that was really good. So there are some nuclear engineers in the YEC community. It's like, unfortunately, Walter Brown has been kind of a outcast in the young Earth community, but the engineers and scientists, he has a following by people like me and said, I think actually your stuff is pretty good. You're not being given credit for it. He was among the first to find out. He's like you and me, Emory. He would go to these obscure journals and just dig stuff up, dig up obscure experiments that are not on the American soil. He found out experiments in Ukraine that was conducted by 125 researchers. So it was a big lab, but it's just written all in Ukrainian, not in English. But he found it, okay? And they're doing electrical experiments where they're taking copper and hitting it with electricity and making all these isotopes. So it's alchemy. You're turning copper into all the isotopes. Oh yeah. Now I brought this when I was at Johns Hopkins and trying to save my grade. I brought all these sort of experiments to my professor. He said, that's the topic of the night. I thought he was going to take my head off. I said, either I'll flunk or I'll get the B and rescue my grade. I got a B. Didn't get the A. But I was glad. But I said, maybe the Lord put it on my heart, put me through that. So I'd worked so hard to do this term paper, and I worked like a dog to dig up all this. I was in the Hopkins Library digging up, and I couldn't believe all the stuff I was finding on electrically induced created isotopes. And there's a lot of quartz in the earth. And when you hit quartz, you get, like your lighters, those butane lighters, it's the piezoelectric effect. When the quartz is hitting each other, it creates electricity. If you're having a lot of tectonic activity, you can create lightning. And you might have enough electricity to create these isotopic things. And it's like, okay, no one talks about this at the creation conferences. The big three creationist organizations, ICR, CMI, AIG, they poo-poo Brown. And I'm like, okay. But this is exciting. Independent of whether you're a YEC, it's pretty cool to do alchemy. All right, guys? Just coolness. Just for the coolness factor, let's just look at this. And now because of the interest in green energy, we're looking into cold fusion, which actually accesses some of this. And Mitsubishi Heavy Industries is being funded by Google, Microsoft, or kind of big high-tech companies that have money to just throw away on fringe research projects, because they've actually been able to affect transmutations now in isotopes using electricity and palladium. They figured out a few things, and this could solve a big problem for YEC. So I'm just like saying, guys, it's a little too early to rule us out. And it will also solve some of the heating problems that Dave McQueen was talking about when they said accelerated nuclear decay. I think he's close enough, but David Neff and some of the sharp guys on the other side of the aisle, the evolutionists, have rightly pointed out accelerated nuclear decay scenarios. There are various forms of them, and one of them, at least a few of them could be lethal. You don't want to go that route. This may help. So finally, Maxwell's equations and special relativity. We had the Hafel-Keating experiment where we had these airplanes with the cesium beam atomic clocks, and they're flying east to west. And they get up to speed. Einstein predicts you'd see changes in the clock speeds. Now, there's something funny there, is that, okay, if you go faster, your clock is supposed to slow down. But there's a funny thing. What if you're assuming that you're kind of like at a zero speed, and relativity says, you don't really know that you're at zero or something. It's all relative, right? Not quite. because the object undergoes acceleration. And if to use a physics term, as you, just like when you're stepping on the accelerator and you're at a dead stop, the longer you step on the accelerator, the faster you eventually reach, of course, within the limits of the car. So we call that integrate, the integral of the acceleration over time gives you your velocity. So that's kind of the mathematical way of describing when you're accelerating in your car. Okay, that's just kind of the abstraction. It seems that the universe seems to give accounting to how much you've been stepping on the accelerator in one direction. And therefore, speeds are not all relative. And I've seen this in the conspiracy of light. And a lot of people are seeing that. And I've wanted to, as I've been tutoring in special relativity, I said, there seems to be like a privileged velocity, like zero. And you could say maybe there's a privileged position, etc. And things aren't actually all relative, even according to Einstein. And so then that
would blow up a lot of people's day.
Yeah. So is it the creationists who are coming up with these objections? No, it's a secular world. These outcasts, like kind of the Richard Sternbergs of physics. And that's what we'll cover. I'll cover it kind of building it from, because we have electrical engineers like Ben Rex. So shout out to Ben Rex if you're listening. I had you in mind as I was building, making this show. We'll start with Maxwell's equations of electrodynamics, which is the heart of electrical engineering. And then we can build special relativity on top of that. And we can talk about clocks and stuff. I think what I was proposing here with this privileged velocity could suggest that there is indeed an ether. And if the ether changes, depending on where you are in space, you could have variable speed of light. And I'll have some comments on the other speed of light models. I have built an interferometer. What I've said is not original to me. I'm not that knowledgeable to do this from scratch. Reginald Cahill and Demjanov and some other researchers have put this on the table, and including Ron Hatch of the GPS committee, government GPS committee. Your GPS will not work without the Hatch filter. And it was the Hatch filter that persuaded Ron Hatch that general relativity needs amendments. So this is not as crank as some of the diehards out there against creationists would want to say. It's crank because you got Ron Hatch. So shout out and thank you to Dr. Sanford for introducing me to the works of Ron Hatch and telling me about his GPS Council work. I knew of Ron Hatch before that, actually, but I didn't know he's on the GPS Council, nor did I know he created the Hatch filter that makes GPS satellites work. So we'll cover that. And so this is more of the throwaway. If I had to ask me anything, it's like, Sal, why are you a yak? This is part of that 10-hour series. So there you have it, guys. And thank you, Emory. I think I told you some things you weren't quite aware of.
Yeah, I learned a lot tonight. I learned a lot. My brain is still trying to wrap its way around a few things. There is a question for you, Sal, from Wesley Coleman Music. That is a question I could not possibly answer. So I'll let you
what was it? Is it this one?
That one, yes, that's the one.
That sounds like Walter Brown stuff, and I'd say everything's on the table right now. You may actually know more of this than I do, Wesley. So I'm sorry I can't answer your question as thoroughly as up to your knowledge level. But this is Emory. Now you know why I said I might be transferring a little bit out of biology. This stuff is just too exciting on another level, and we do have very capable people like Richard Sternberg to kind of help the ID case along. It's just a matter of finding these guys. I mean, what he did on that RNA in the mouse eye, I mean the DNA in that's. Why hasn't any yet thrown this on the table in a debate, even myself included, when I debated Daniel Stern Cardinal, I could have thrown that one out. I just didn't have time.
Mm-hmm. Yeah. Well, if I ever do a debate, Sal, I'll make sure I bring that up. Yeah. So,
any closing thoughts, brother?
I mean,
thank you.
tonight, I think.
Yeah.
Go ahead.
No, I'm glad to see you healthier. You were kind of worn out after a day of work, and then you had 2 hours of me.
Yeah, yesterday I was crashing. I was whipped. Much better today. So I think anybody who watches this stream will definitely get their money's worth out of it. I mean, it's free, so of course they'll get the money's worth. But anybody who listens through this stream will learn something. I think even the most uninitiated to the scientific realm can still pick some things up here, because we went through some stuff that was really techie and some stuff that was pretty straightforward and understandable. I think there was a lot of good stuff here, and I think that it was very helpful. And I appreciate you, Sal, letting me come on and talk about it, and read through some stuff with you. And we'll see how it goes. Hopefully I'll be able to catch your physics stream that I will understand none of.
Sounds great. So God bless you all. And if you don't see me on streams on this channel, there's going to be some streams in the next couple of days or so on other channels, Dr. Dan's channel, and I'm tentatively scheduled for the non sequitur channel. So we'll see how this, I don't know, it's not been confirmed yet, but we'll see how this plays out. And I don't think they would mind me announcing that, because they would love all the advertisement they can get.
Well, NSQ already has the video posted up on their channel, the upcoming event. So.
Oh, we're on the hook. You saved me, Emory, because I was waiting for the 11th. Oh boy, I'm in trouble. So thank you. The Lord sent you my way. So guys, I may appear then on the non sequitur channel, and then I'll be with Dr. Dan on the following day, and who knows where else. So guys, take care, and God bless you. Have a great evening.
Have a great evening, everybody.